Okay. So in 2021, 47,412 hematopoietic stem cell transplantations were performed. According to estimations, this number goes up 10 to 15% each year. And despite improvements in prophylaxis and treatment strategies, 30 to 60% of patients develop an acute graft-versus-host disease. And with this, I want to welcome everyone who found the time to join this webinar. My name is Anastasia. I am a PhD student in the lab of Rika Harasti at the University Hospital of Tübingen. And on this part, thank you so much to the OTS for giving me the opportunity to present my work about how we can use siRNAs to fine-tune the immunity and thereby to enhance the efficiency of cell therapies. So in my talk, I'm going to give you a short introduction about graft-versus-host disease and graft-versus-leukemia. I will present you my recent and most exciting results. And at the end, I will highlight the key points and explain you my future plans. So let's dive in. So allogeneic cell therapy is the most potent tool to eradicate leukemias. However, allogeneic T cells can also trigger the graft-versus-host disease, which is a severe condition that can lead to death or develop into a devastating and chronic form. Unfortunately, intracellular mechanisms driving GvH or GvL effects are not completely understood and remain largely unexplored. The current GvHD drugs cannot differentiate between GvH and GvL effects, and leading to leukemia relapses and infectious complications. And we believe that siRNAs may have the potential to differentiate these both pathways by fine-tuning the phenotype of donor T cells and thus preventing the development of GvHD while preserving the therapeutically essential GvL effect. So the chemical structures of our siRNAs features a 21 nucleotide long antisense strand and a 16 nucleotide long sense strand with a cholesterol conjugation on the 3' end and every ribose being modified. The phosphatidated tail facilitates membrane association, and the modifications with 2' O-methyl and 2' fluoro enhance the stability of siRNA and decrease the activation of the innate immune system. All of our siRNAs are synthesized and provided by the Quarva lab, and I want to thank all of the involved group members for making our research possible. So in drug development, one of the most important decisions right at the beginning to make is to choose the right targets. So on the next slide, I want to present you 4 targets that my work is focused on. So let's start with VAPAL. VAPAL's role is to regulate the chromatin architecture. And his novel role in GvHD emerged when Sun and colleagues carried out analysis of microRNA differential expression in allogeneic T cells. They used a novel approach to identify differentially expressed mRNAs by co-immunoprecipitating Argonaute-bound microRNAs and mRNAs. They identified a network of microRNAs that were downregulated specifically in allogenic stimulated T cells. And they showed a group of mRNAs, including VAPAL, predicted to be targeted by these microRNAs. They have also shown that the downregulation of VAPAL can improve the severity of the graft-versus-host disease in a mouse model. And another group performed transcriptome profiling of primate T cells during acute graft-versus-host disease, which has led to discovery of signaling pathways that, when inhibited, ameliorate the disease. They discovered pathways that are specific to alloreactive T cells and thus to GvHD. And they found that transcripts as RUN, AORCA, and KIF that are usually associated with the mitotic cell spindle were significantly and specifically enriched in T cells during GvHD. So we have designed 11 or 12 siRNAs for each target, and they performed a screen in epithelial adenocarcinoma cells in HeLAs, in Burkitt's, which is T-cell leukemia, cell line, and also in primary activated T cells. And the screen data show that the leading siRNA candidates correspond mostly throughout the cell types. However, you see differences, for example, here in AURCA, as well as in KIF15. And these differences may be due to cell type specific mRNA localization, SNPs, or maybe splice variants, for example, like intron retention, which is quite prevalent in T cells. So after identification of the siRNA leads, we tested the silencing efficiency and activity of the siRNAs of different concentrations in the cell types mentioned previously. So HeLa, Jurkat, and the primary activated T cells again. And interestingly, we observed an 186-fold differences in the potency when you look at the AURCA IC50 values, so between the cell types. And when looking at the highest concentration, which was 5 micromolar here, we were able to achieve 55 to 95 silencing activity in HeLa cells, 55 to 85 silencing activity in Jurkat cells, and 65 to 72% of silencing activity in the activated T cells. And if you take together the silencing efficiency at the highest concentration, together with the IC50 values, it demonstrates a decrease in siRNA efficiency towards the primary T cells. But very surprisingly, the VAPAL siRNA shows quite the same silencing activity in HeLa as well as in Jurkat cells. And why is this surprising? Because HeLa cells are expected to have a much better siRNA uptake compared to Jurkat cells. And so possible explanations for these discrepancies are, for example, the uptake efficiency, which I just mentioned. So, for example, there is higher in HeLa cells and less efficient in Jurkats or in the primary T cells. Another explanation would be the RNA availability and also the splice variants. So one approach to analyze the RNA availability inside of the cells was to perform subcellular fractionation experiment here and to quantify the mRNA amount in the cytosolic and nuclear fractions. And these experiments revealed that up to 30% here in HeLa of mRNA are localized in the nuclear fractions and are thus not accessible to siRNAs. However, the silencing efficiency was the best in HeLa, meaning that these results do not explain our 186-fold difference in silencing potency. So we hypothesized that localization to compartments in the cytoplasm, such as endosome or lysosome, are responsible for the lower susceptibilities to siRNAs. So after testing the silencing efficiency of siRNAs, I wanted to test if our siRNAs are able to inhibit T cell proliferation upon allogenic stimulus, and thus the development of graft-versus-host disease while preserving the graft-versus-leukemia effect. So therefore, I tested 4 different concentrations of siRNAs in a functional GvHD assay. I used 2 different stimuli to activate the T cells. So the non-specific stimulus is where the T cells are activated by non-specific activation beats. In the allogenic stimulus, the T cells are activated by major mismatched dendritic cells, which is the common method for an in vitro GvHD assay. And what you can see here, very good, is that the only siRNA showing a robust and efficient T cell inhibition is run, but upon both stimuli. So here, upon non-specific stimuli, we have an inhibition up to 60% and up to 80% upon the allogenic stimulus. So yeah, the VAPAL and AURCA and KIF targeted siRNA, they show only a moderate effect in allogenic stimulus and no effect in the non-specific stimulus. And very interesting here is that the data show a broader distribution of the inhibitory effect in the allogenic stimulus. because here you see it's quite stable and here you have a broad distribution. So this actually reflects quite well the situation in the clinic, where we have a broad distribution of patients' response to the GvHD therapy and prophylaxis, which makes the therapeutic outcome unpredictable. So precision treatments preserving non-specific T-cell activations are needed in the clinic. And therefore we wanted to further optimize the siRNA treatment in order to make it more specific to the GvHD situation. And our first approach was to formulate siRNA into mesenchymal stem cell derived extracellular vesicles, because the mesenchymal stem cell derived EVs were proven to have an intrinsic therapeutic effect on GvHD. And we hoped to use this effect to potentiate the efficiency of our siRNAs. So I tested extracellular vesicles with and without VAPAL siRNA in the functional GvHD assay regarding the ability to inhibit T-cell proliferation. And indeed, the effect of VAPAL siRNA could be enhanced up to 50%. Upon the allogenic stimulus, and the non-specific activated T cells were not affected. And here again, you also see quite a broad distribution. And this again may be due to the metabolic state of the T cells itself, and also due to the batch-to-batch differences of EVs. And we also plan to formulate other siRNAs into EVs, for example, RUN. And we hope that the effect of RUN will become more specific to the GvH situation and more specific to the allogenic stimulus. The second approach to enhance the inhibitory effect of siRNAs was to combine siRNAs targeting different genes. So we combined 3 siRNAs targeting one of these genes. And then I tested again these siRNA mixes, all 4 combinations in a GvHD assay. So very surprisingly, we have here the mix 1 that inhibited T cells up to 72%, and mix 2 that inhibited T cell proliferation up to 52%. And the non-specific T cells were not affected. Here again, you can see quite well 2 populations. So this is actually not due to measurement issues, but we believe that this is the biology. So you have a population where the siRNA treatment works very well, and then you have another population where the siRNA treatment still works, but it's less efficient. So as it's the same with the patients in the clinic. So we went on and combined 2 siRNAs, and what we saw was that the exchange of only 1 siRNA changed the effect up to 2-fold regarding the T cell inhibition. So these results indicate that specific genes regulate the T cell activation or proliferation in a synergistic manner. And here, again, due to the broad distribution of the inhibitory effect on the allogenic stimulus, more repeats are needed. But still, the current results indicate that the T cell inhibition of the siRNA mixes is stronger than the one of the pairs. So, to further understand the underlying mechanisms of siRNA mixes, I analyzed the silencing of each mRNA in the mixes of T cells with different activation states. So I tested resting T cells here and beat-activated cells here in the second row. And what you can see is that the silencing efficiencies differ between particular genes and also between the activation states of the T cells. So for example, the inactivated T cells, you have in general a bit less silencing compared to the resting T cells. Also, KIF15 silencing seem to be less potent compared to the other siRNAs in both cell types. And very interesting and surprising is that if you look at the activated T cells and the Vapalas siRNA, it seems that the silencing depends on the mixes. So here we have around, in the mix 1, we have around 24% of silencing for Vapal, but we have 65% of silencing of Vapal in the mix 3. So yeah, this data is quite new. I think it's from last week. So the repeats are needed, and I also need to analyze mix 4, which is completely missing here. And I also want to compare these results to a third kind of T cells, so allogenic activated T cells, which I co-incubate with dendritic cells. So after showing you that we are able to inhibit the T cell proliferation, we wanted to test if our siRNA treatments disturb the therapeutically essential graft-versus-leukemia effect of the T cells. And therefore, I have established an GvL in vitro assay with human cells, where I co-incubated major mismatched dendritic cells with T cells and with an acute myeloid leukemia cell line, MOLM13. And we observed a reduction of the alive leukemia cells compared to the untreated, and an increase of dead leukemia cells when co-incubated with T cells and dendritic cells, which reflects an efficient graft-versus-leukemia effect. Also, treatment with our siRNAs mixes did not lead to an increase of alive leukemia cells, indicating that the GvL effect is preserved. In the second graph, you can see that the number of alive T cells slightly increased in the siRNA-treated samples compared to the untreated, indicating that the siRNA treated cells still have enough fitness to eliminate leukemia cells. And an even better model for a GvL assay would be to use patient-derived leukemia cells instead of the cell line that we are currently using. And yeah, we can't wait for the ethical approval to start these experiments. So after showing you the most exciting results that I obtained in the 1.5 years, I want to highlight the key points here and to explain my future plans. So our most potent mix, consisting of Vapal, RAN, and AORCA, defines a pathway within the T cell selective to GvH. So for a better understanding of the mechanisms, I want to perform an RNA-seq as well as T cell phenotyping via flow cytometry. And I hope that this will help us to understand the mechanisms underlying these mixes. And what I was also able to show you is that the efficiency of siRNAs really depends on the T cell activation status. And therefore, I want to test a few more triggers for activation, such as CMV or, as I mentioned before, the patient leukemia cells. Also, we have proven that there are pathways that are specific to GvH. And the upcoming question is if these pathways differ in the individual donor and recipient pairs. So this raises the question whether a personalized treatment may be the approach of choice in the future. And also, we suggest that if you screen siRNAs for T cell indications, that you should try various T cell models and not only the normal cell type for screening like HeLa. And also another very interesting question is to test if the protein silencing actually correlates with the mRNA silencing. And I am establishing the Western blot for these genes and proteins. So yeah, this is in progress. And yeah, we have come to the end of my presentation. And I want to thank my supervisor, Rika Harasti, for her outstanding mentoring. I want to thank my colleagues, Tatiana and Xavi, and all the other excellent scientists that contributed to the success of this work. I hope you enjoyed my presentation, and I would be happy to receive questions and yeah, maybe exciting ideas for new experiments.
Yes. Thank you very much, Anastasia. I think it was a super clear presentation. So time for questions. If you have a question for Anastasia, please use the Q&A function so then we can read the questions. Maybe to start with a question from my side. At the very beginning of your presentation, and I think you also came back to this during the discussion, you are using 3 different cell types. And you mentioned that the effect that you measure is different in all these 3 cell types. It can be SNPs, it can be different expression levels, there can be intron retention, different splice variants. So what would be the best cell type to do the screen? Or is it necessary to do the screen in 3 different cell type? Or should you say, okay, maybe just only do the screen in the T cells?
Thank you. It's a great question. So I think it really depends on what your target cells are. So for example, when your target cells are immune cells, then I would definitely try to do the screening in some kind of immune cells or even primary cells. I know it's much more work, it's more expensive, but as you can see, there are big differences between the cell types. So for example, you do a screen in HeLAs and you decide to choose one as siRNA. This is something actually that happened to us. And then you do just out of curiosity, you perform the screen in your target cells and the T cells, and then you see, okay, but another siRNA performs better. So you made the wrong decision. So that's why I would really recommend to do if possible, to do the screen in your target cell types.
Yeah, so for a very first screen, just to see whether siRNAs are effective, yes or no, would it be sufficient to do inhaler cells and then to look at more at the effect at the more difficult cells?
Yeah, so I think if you just want to check if your siRNAs are working, then I think that HeLAs are perfectly fine. But if it comes to really choosing the right siRNA candidate, then maybe it's beneficial to use your target cell type.
Cool, thanks. I think we move to the Q&A section. Just see, we have a few questions. So first question is from Stefan. He actually has 2 questions. So first question, could you elaborate how you would translate your results into a clinical application?
Yes, thank you for the interesting question. So the common issue with siRNAs or RNA therapeutics is usually targeted delivery, right? So I think everyone knows. But we can, I think, undergo this problem. So for example, when you have the cell therapy or when you have the cells that you want to infuse into the patient, so right before giving it to the patient, it would be an option to treat these cells with like a special mix, for example, of siRNAs, and then you give the treated cells to the patient. This means that you automatically have the targeted delivery because it cannot go anywhere else, and you still have the effect, and hopefully you can prevent the development of the graft-versus-host disease.
Yeah, would that be difficult timing-wise? Because I kind of know that you need a few days to have to see the effect of the downregulation.
So we also performed some kinetic experiments with siRNAs, and for the uptake, 24 hours are more than sufficient for the uptake. Until you see this downregulation of mRNA, depends on the cell type. So we have cells where, for example, for the HeLAs, 3 days of incubation was enough to see a mRNA downregulation. But in Derkit cells and the T cells, 6 or 7 days were better or necessary. So yeah, and we are also testing if you can freeze the cells with the treated siRNA and then you thaw them again. So that's also point of research that we are focusing on.
continue. The second question from Stefan. That was a more practical question, so I think you can answer this relatively quickly. How did you pack your siRNAs in the EVs?
So I think the protocol is just to, I mean, you have to calculate the right amount of the EVs, and then you incubate it. I think it was 1 hour at 37 degrees. So just the EVs and your siRNA. And then you have to do a centrifugation, ultracentrifugation for, I think it was 90 minutes and 100,000 G to remove the siRNA. And yeah, and then you can use the EVs right away.
That sounds okay to do. Question from Rebecca. Nice work. I think a similar question as we had before. If T cells are rapidly dividing, are you concerned about dilution effect of siRNAs over time? Have you measured that? And what kind of durability will you need for therapeutics?
Excellent question. Actually, yes, we performed long-term experiments where I have treated T cells with siRNA and incubated them for up to, I think, roughly 28 days. And then I measured the silencing efficiency. And so we were able to achieve silencing efficiency up to, it was like up to 10 T cell divisions. Because T cells, they cannot proliferate as much as they want. Usually the CD4 cells undergo around 7 to 8 divisions, and CD8 T cells undergo 7 to 8 divisions, and the CD4 T cells undergo around 10 divisions. So, and we were able to show the silencing activity in this time period.
Thank you. So we have time for one more question, one short question. So if you have more questions for Anastasia, I think it will be okay to use the chat function. And Anastasia, if you want to answer the questions in the chat, that would be helpful for the participants. Yes. So last question is, when analyzing the mixes, it looks like mix 2 has a stimulatory effect for one of the two subpopulations. Furthermore, some of the tested genes seem to be a bit repressed.
Okay, I think Elena is referring to this graph. So yes, it looks like an upregulation, but I actually think it's not an upregulation. It's just that some populations have no effect. So I don't see real upregulation of T cell proliferation. I have never seen this.
Okay, well, thank you. So that thank you very much for your excellent presentation and excellent questions and answering of the questions. And it's time to go to the second speaker.
Great talk. So our next speaker is Linda Popella. So go ahead, Linda.
Yeah, also warm welcome from my side. It's a pleasure to present the data from the poster from last year's OTS. And I hope you can see the slides properly. It's just a little bit slow today, sorry for that. Okay, so now we are completely switching the field. So now we dive into microbiology, and we envision the usage of RNA antibiotics as novel antibiotics, novel class of antibiotics. But why do we need novel antibiotics? So I think we are all aware of the increasing emergence of antibiotic resistance among bacteria, and it's estimated by 2050 that 10 million deaths will be attributed only to antimicrobial resistances, and this will be thus the leading cause of death worldwide. And apart from antimicrobial resistances, we know that our human body is populated with a plethora of different species of bacteria. And I just want to have the laser point now. And we know that there are some commensals, so they are good for our health and immunity. But commensals can also turn into pathogens and can there be a trigger for cancer or autoimmune diseases, and they are the purely pathogens that are only bad for our health. So what we envision is to develop programmable antibiotics that can selectively edit the microbiome. And we are using peptide-coupled antisense oligomers in our lab, and we are particularly interested in PNAs, so peptide nucleic acids, as the backbone modality. And in the bacterial field, those azoles are in the range of 9 to 11 nucleobases, so shorter than for the mammalian field. And we are also conjugating those azoles to certain molecules to facilitate intrabacterial delivery. And commonly, cell-penetrating peptides are used for this, and they can thereby facilitate the uptake of the PNAs into the cytoplasm of the bacteria. And we are targeting the translation initiation region of certain bacteria genes to block ribosome binding and thereby to block translation. In the case of an essential gene, we can thereby block translation of this gene and elicit thereby growth inhibition of the bacterium. But what is a good susceptible essential gene target in bacteria? So there hasn't been like a systematic study so far to know what is the best essential gene to target and to inhibit growth of a certain species. And this is why we did a small screen in a recent study where we used uropathogenic E. coli as the model organism, which is one of the leading causes of urinary tract infections, which can be chronic and they can also be recurrent, so they're a huge problem in the clinics. And there we have tested 11 different essential genes, which were selected based on different expression levels to also see whether high or low abundant transcripts are better or worse in targeting with the PNA and in terms of growth inhibition. And I only want to mention that ACPP, so it's the SI carrier protein, which is involved in fatty acid synthesis, is used as one of the gold standards in many bacteria as it is a quite susceptible essential gene when it comes to PNA inhibition. And when we were analyzing those 11 different genes, we didn't come up with the real predictor of what makes a good PNA or what is a good essential gene target. So we didn't know so far what are important features of efficient PNAs or what is a good susceptible PNA target in terms of essential genes. So this is why we decided to go for an essential gene-wide screening, and we therefore used previous publication to select essential genes, and we took in total 293 essential genes into account, and we designed 2 PNAs of 9 nucleobases to target the translation initiation region of those essential genes. We conjugated those PNAs to the cell-penetrating peptide KFF, which has been shown to be a good delivery vehicle for the intrabacterial uptake, and it doesn't result in a huge stress response. So we can be sure that really only target gene depletion is the leading or is the cause for growth inhibition, which will be observed later. And so our library was roughly 600 PNA sequences, and we tested all those PNAs in a lab strain, the E. coli K12, the BW strain, and also on the clinically relevant uropathogenic E. coli 536 strain. And what we did first, we tested the PNA library at 10 micromolar for the 2 strains. And this is just an example of the data that we acquired. So we monitored the growth behavior and the presence or absence of the different PNAs. And then we had a readout after 24 hours to see whether we completely inhibited growth or whether growth was observed in the end. And a simple visualization of the 24-hour data is given in this diagram here. So on the left side, we have growth inhibitory PNAs, either in UPEC or in the K12 strain, or overlapping with both organisms, or we have the PNAs that did not elicit growth inhibition in either of the 2 strains. So what we can say in the end, that the majority of PNAs were able to inhibit growth in UPEC or K12, but UPEC seems to be more susceptible to PNA treatment than the K12 strain. And in total, we had roughly 70% of all PNAs that inhibited growth in UPEC, only 40% did so with K12. And this corresponded to roughly 80% of all targeted genes in UPEC and roughly half of the targeted genes for K12. And one possible explanation why some PNAs were not growth inhibitory could be that we only achieve knockdown, but not the complete knockout, which is the basis for an annotation for an essential gene. And maybe some residual protein levels could still facilitate growth. So what we concluded from here is that most of the PNAs were growth inhibitory, but there are also strain-specific differences in susceptibility upon PNA treatment. And here we think that it may be due to the LPS composition of the two strains. We then analyzed several features of the PNAs included in our library, and this is the first analysis that we performed. So when taking the growth inhibitory PNAs versus the non-growth inhibitory PNAs, and two of the top features that really discriminated between growth and non-growth inhibitory PNAs were the melting temperature and the off-target range of the PNAs. So melting temperature totally makes sense. The higher the melting temperature, so the more stable the PNA can bind to the target mRNA, the better they are. And when it comes to off-targets, and here we are looking at total off-targets, so anywhere in the transcriptome or genome, this can then mean that there might be a sponging effect. So if we have more off-targets in the bacterium, the PNA will be also binding to those off-targets and do not reach in sufficient amounts the actual essential gene target, which means that the PNAs might be less efficient. In the end, we also performed some machine learning analyses to find the top predictors for PNA efficacy. And what we learned from here was that the melting temperature is the top feature among all analyzed. And what we see here is the SHAP value, so how much a certain feature is associated with the model prediction, and then also the feature value, so a high feature value, for example, for the melting temperature. So high melting temperature means better PNA efficacy in this case. And the second most important feature was self-complementarity, which is also quite logic that the more self-complementary a PNA is, the less efficient the PNA can bind to the target mRNA because it will be sponging itself away. And then the third most important feature was the off-target within the translation initiation region of other genes. And this is now different to off-targets total, because here also the translation initiation region of other genes will be bound. So translation will be blocked off off-targets, and the more off-targets in the translation initiation region are present in the bacterium, the more likely the PNA will elicit growth inhibition. And going a little bit further into finding very susceptible or highly susceptible PNA targets, and also to gain more insights into strain and gene-dependent differences, we performed a sub-screening of all the growth inhibitory PNAs identified before. So we went down to 0.3 micromolar now, and we did the screening again in K12 and UPEC. And what is depicted here is the final OD after 24 hours. In red, growth, and blue means no growth, from high to low PNA concentrations. And again, we observed that UPEC seems to be more susceptible than K12. And when we projected this complex data set into a violin plot, we see that for UPEC, we have a nice separation into the different MICs from 10 to 1.25 micromolar, whereas K12 seems to be less susceptible, and most of the PNAs were not growth inhibitory until 10 micromolar, or they had an MIC of 10 micromolar. And what was interesting, that we identified ACPP as the most susceptible gene target, which was nice to see that we identified the gold standard as one of the most susceptible gene targets, but we also found additional ones, which I will come to later. So we then took this data set from the sub-screening from 10 to 0.3 micromolar, and we performed, again, bioinformatic analyses to have a closer look into the features already analyzed before, but now with the differentiation into the different MICs. And this is just a sneak preview of what we analyzed. So we analyzed several different features, and these are the most important ones where we saw significant associations with the MIC. So again, melting temperature and self-complementarity were the top features that were associated with the final MIC. And what we also found is that the gene expression seems to be slightly correlating with the MIC. So the top susceptible genes, they have or in general, a higher expression level than the genes that are not susceptible, which could also be correlating with their biological function. So highly expressed genes are more important or more vulnerable than the low abundant genes. Relative fitness is a factor which comes from a CRISPR-I study that was published in 2020. And the lower this value, the more susceptible the gene was in the CRISPR-I screening, which is also correlating with our data. So lower relative fitness, the more susceptible the gene is. We also identified the purine percentage to be associated with the MIC. So the more purines in the PNA sequence, the less efficient the PNA seems. And also, if there are essential genes upstream of our gene target, is an important feature, and this is associated with the final MIC. We are also looking into KEC pathway enrichment of the genes. So if we find some pathways that are more susceptible than others, and this will also be interesting to see if there is a difference between the K12 and UPEC strains. And we then also performed machine learning again with the UPEC data set with the different MICs, and we find similar top features as I showed before. So the melting temperature and self-complementarity are the two top features that predict the PNA efficacy. And as mentioned on the previous slide, the vulnerability based on the CRISPR screening expression level. And we also see that the off-targets in the translation initiation region of other genes is associated with the final MIC. So this gives us now a good guideline of which features have to be taken into account to design a very potent PNA that can inhibit growth. We then also wanted to validate the MICs that we gained during the screening, and we selected then interesting PNA candidates for further analyses. So as shown before, we performed MIC assays, and we took a look at the final OD after 24 hours to define the MICs. And this is just a summary of the MIC assays that we performed, and we went from 96 well plates to 384 well plates, which is now good that we can screen more PNAs in a shorter time period, and we can also screen a larger panel of enterobacteria, which I will come to on the next slide. And what we have seen, we could validate all the MICs, and we have identified PNAs that are very potent in both strains that we have analyzed, and some PNAs that seem to be more potent in growth inhibition for the pathogenic UPEC strain, which is also quite interesting to see. So as mentioned, we then also tested those PNA candidates on a broader panel of Enterobacteriales. So not only UPEC and the K12 strains, but also a probiotic strain. And we have identified thereby ACPP, as mentioned before, as the top gene among the essential genes, and also some additional ones that have to, or that seem to have a good activity against those tested ones. And we also verified the more strain selective PNAs, like those two highlighted in green. And apart from UPEC 536, we analyzed clinical isolate of colorectal cancer and another UPEC strain, and also Salmonella or one Klebsiella strain, to see whether we have some more strain or species selective PNAs or pan PNAs that could work against all strains. And we found that ACPP seems to really work quite good in the tested bacteria, which wouldn't be a very good precise antibacterial agent, but it could work as a more broad spectrum antibiotic. But we also identified some PNAs that could really be specific against a certain species. And this could also lead to the development of species-selective RNA antibiotics, such as this one here, where we found mismatches in the target sequence in those organisms, and we could then design species-selective RNA antibiotics. And the next steps will be to perform RNA sequencing in the after treatment with the PNA candidates that we selected before. So in both bacteria after different time points, and our goal is then to find out if the PNAs really selectively deplete our target gene and whether we have some off-target effects or not. That could also lead to growth inhibition. We also want to get insights into the induction of stress response pathways in the two bacterial strains that we were using, and also to find out if there are similar kinetics of the induction of stress responses triggered upon the presence of the different PNAs. And we would also like to identify the mechanisms that lead to growth inhibition, and if they are shared between different PNAs that target different genes or different pathways, or if the mechanism of growth inhibition is target gene dependent. What we also identified within our screen and based on the results that we gained is that we see that the PNAs, the two PNAs that we designed per gene, were not always both leading to growth inhibition, but sometimes only one of two PNAs per gene were growth inhibitory. And this is a simplified graph to illustrate this. So the left panel shows the UPEC data, the right panel the K12 data, and on the y-axis we have the roughly 300 target genes. And this is showing the different MICs that we found, so 1.25 to 10. And here, for example, we see that, again, ACPP, so both PNAs for ACPP were growth inhibitory in UPEC at a very low MIC. And in K12, the MIC was a little bit higher and twofold different. But we also identified some PNAs or some target genes where the two PNAs had quite different MICs in the end, and we want to know what makes the difference between the two PNAs. So is it dependent on the target sequence within the gene, or are they different because one of those has an off target that is then the trigger for the growth inhibition? And we could find out more about those differences when performing sequence tiling for the target genes. And what does sequence tiling mean in this respect? So sequence tiling means that we walk along the transcription initiation region, and we can also say that we do like a catwalk. So the AUG means CAT. And we walk along from -14 to +12, and we design PNAs for all the different target regions and analyzing the final OD after treatment. And when we did so for our gold standard gene ACPP, we found some hotspots along this translation initiation region, and some PNAs didn't work that good. And this would be quite expensive when we would order all of those PNAs from the company that we are working with. So we have a collaboration now in-house where we can synthesize the PNAs on our own. And we saw a similar distribution of the final OD when comparing the company-derived and the in-house derived PNAs. And so we did now these sequence tiling experiments for some gene candidates, so essential gene candidates that we discovered before. And we found that there are quite huge differences among the different essential genes. So we have some PNAs that seem to work better than others. And this is not always the same position for each single essential gene. So in the end, we want to then look a little bit further into the target sequence and whether the target sequence tells us about the melting temperature and self-complementarity. These are really the two most important features that then can discriminate between working and non-working PNAs, and also if we can use our machine learning algorithm to predict the MICs and to then back compare with the experimental data if our prediction is good enough. And what else we did with the screening data is we then selected known drug targets for PNA targeting and to define whether those already known or applied drug targets are also good PNA targets. But what we see here on the right side, they are not really good PNA targets in all cases. Only MORA seems to be an medium good drug target. So targeting the RNA is something completely different than targeting the protein, of course. But we can also then find maybe novel PNA sequences for known drug targets to develop good novel antibacterials. And to sum up our PNA screening, so what I've shown in the beginning is that most of the genes are druggable with PNAs in both strains that we tested. We identified the top features to predict PNA efficacy in UPEC, and we identified some novel susceptible targets with differences among strains and different species. And what was surprising is that we identified ACPP among the top candidates in both or all the strains that we tested in the end. And we are highly interested in why this is the case, if it's just because of its function during fatty acid biosynthesis, or if it's something associated with the sequence of the PNA. And I also want to give a broader outlook on what else can we do with PNA, so we can not only selectively deplete pathogens or rough commensals, but we could also resensitize bacteria to already used antibiotics. We can also suppress certain pathways to inhibit the drug inactivation by some commensals so that the patient gets again sufficient amounts of the drug. And we can also target bacterial viruses, so phages, to modulate infection processes, and which could be also important for biotechnology. And with this, I want to thank you for your attention. And of course, I want to thank our boss, Professor Jörg Vogel. I also want to thank our collaborator, Lars Barquist, and Jakob Jung and Tao, who did a main part of the work in this project. And if you have further, or do you want further information, you can contact us via these email addresses. So I'm open for questions. Thank you.
Yes, Linda, thank you very much for this very clear presentation. I think it's a very nice application of PNAs. So what would be your favorite strategy? Would you go for a strain selective target so that you can really specifically target one specific strain, or would you go for a more general approach that would be applicable for many different kinds of bacteria?
So since we have the tool in hand, my favorite application would be really strain or species selective application. But in the end, all that matters is to have something in hand if none of the conventional antibiotics would work.
Yep. Of course, and it probably is a very clear question. So how would you turn this into a clinical application? How would you deliver these PNAs to the place where the action should take place?
Yeah, so this is always the big question of delivery. So we are also currently working on some strategies to deliver them in a tissue-specific manner, for example. And I'm mainly focusing on urinary tract infections. And here we can also think of some flushing of the bladder to get into the bladder, and then the PNA has to stick to some epithelial cells and then also get internalized to the cells or the umbrella cells, because UPEC is also an intracellular pathogen. So here we have a further complication to not only treat extracellular bacteria but also intracellular bacteria. And for gut pathogens, it will be even harder. For the lung, we can think of some sprays that will be inhaled and then to get into the lung. But it's really a huge, or it will be a huge amount of work to really get into tissue-specific delivery. And
yeah, I think this is probably this will be the challenge, right? Because you have a few very good targets. It might be quite challenging to have this tissue or cell specific. So let's move to the questions in the Q&A. So please, when you have a question, use the Q&A. You can also use the raise hand function, so then you can ask the question yourself. So first question is, why did you decide to use 9 nucleotide PNAs? Because obviously, if you change the length, then also many of the other the TM will change, the number of self-complementary bases will change. So a lot of other of these parameters will change.
So the most important thing why we decided to go for 9 mers is the fact that, so usually 10 mers are used for bacteria, or 10 to 11 mers, but the bigger the molecule, the less efficient the uptake will be, as there is no way of endocytosis pathways like in eukaryotic cells. So we really depend on cell penetrating peptides that form some pores, or there is some other pathways that we do not fully understand to really deliver the PNA into the bacteria. So we have this size limitation with the PNA, and in this previous publication we compared 8 to 11 mers, and we saw that 11 mers are less efficient than 10 mers, and 9 mers really have the same or almost the same activity as the 10 mers, and they should be still very selective in the target inhibition and not having that many off targets, as the genome size is way smaller for bacteria compared to mammalian cells.
Yeah, I was also wondering about that, about the off-target effects, because the clinical application is in human beings, right? So should you also focus on the off-target effects, or maybe on-target effect in human beings? Because I think the studies that you did so far is mostly focused on the bacterial genome or transcriptome.
So yet we are only focusing on off-target effects in bacteria, but of course we also have to be careful with off-targets in eukaryotic cells. So if only treating extracellular bacteria, we can somehow avoid some off-targeting in mammalian cells due to formulation or encapsulation or something. But when treating intracellular infections, we have to be careful with that.
Fully understand. Question and a remark from Rebecca. such an interesting application of PNA to target discovery. Could the correlation of PNA efficiency be related to the rate of translation of the target mRNA as well?
So we have taken a previous study into account, which gave us mRNA half-life and translation efficiency values, and we did the analysis with our previous screen with the 11 essential genes, and we didn't see a clear association. But we want to have a closer look with now this huge data set again into all those features. So now we took, I think, in total 15 features, but we have already a list of more features that we are going to analyze with our MIC data. But that's a really good point, yeah.
Yep. Last question/remark, maybe you can comment on it. Rebecca again says, bacterial blood sepsis would be interesting. Do you agree?
Would be interesting, yeah, of course. Yeah. So there are so many applications we can think of, and especially when it comes to these antibiotic resistant infections.
Yeah.
But yeah.
Really agree on that. That was the last question in the chat, and I think it's also around the hour, so I think I give the word to Erin.
Great. Well, thank you everyone for attending today. Just a reminder that the webinars are recorded and posted on the website. So if you've missed any past webinars you're interested in watching, you can check it out there. And if you really enjoyed this talk, you can share it with your colleagues. We also have the second part of the award spotlight series coming up in February on the 29th. Same time, another Thursday at the exact same time, and we'll be featuring 2 more speakers. If you're interested in presenting in the webinar series, please do get in touch with us at info@allogotherapeutics.org. You can contact us directly as well, and we would love to have you speak. So thank you so much again for our speakers today. You both did an excellent job, and it was really engaging. And thanks for the great questions as well. Have a great day, everyone. Take care.